Turn on your video.
Yes, of course.
Alright. Okay.
Right.
So we can start the session, and post that we'll have the Q&A. Would you like to share your screen?
Of course, yeah. All right. There we go.
Yes. It's all yours.
Great, thank you. Okay, so I'm Dina Zielinski. I'm a bioinformatics researcher currently living in Paris, France. And I'm going to talk to you today about how we can store digital data in DNA. So I'm a bioinformatics scientist, and most of my job entails working with files that look like this. Now, if you can see, these are pretty scary looking files that actually have tens of millions of lines oftentimes. And so I often get emails from our administrator asking me to please delete some of my files once I'm done analyzing my data. So this is a basic sequence alignment file, and we're constantly, as scientists, at exploring this tension with where we can actually store all of our data and also be able to analyze it without having to delete it. Even in the past, we've thought it might be better to simply delete sequencing files and redo the sequencing process because it's just too costly to store all of this data. So on a more relatable level for most of you, we generate a lot of data every single minute as human beings all around the world. Every single minute, there are 4 million videos streamed on YouTube, tens of thousands of photos on Instagram, and many hours streamed on Netflix, hundreds of people joining LinkedIn, millions of emails sent through Gmail, and few 100,000 tweets, maybe even more so, especially given recent unrest.
Excuse me, Dr. Dina, can you full screen your presentation, please? Can you full screen your presentation?
Yes, how's that?
Yeah.
Great. Okay, so we generate a lot of data, and most of us don't really think about where this data is going, right? I send out my tweet, I send out my email, but really everything that all the data that we generate, all the data that we store, it's not simply in this mystical unknown server. It's actually stored on these server farms, which are highly energy consumption. These consume a ton of energy. They're very costly to maintain. Not only do you have to maintain the hardware, which you're constantly reading and writing to, but you have to cool these hard drives. And so this is where all of your tweets, all of your Netflix videos, this is where everything is in these giant server farms. So this is actually a server farm in Sweden that belongs to Facebook. And you can see it's pretty big. And the reason why Facebook chose Sweden is not necessarily because of cheaper real estate costs. It's actually because it's so much colder up here that it requires much less energy to actually keep all of those servers cool. The problem is we're running out of devices to store all of this data, which I will soon get into. So most of us, if we were to see this inbox empty, it would be kind of a relief. I don't know about most of you, but my inbox is usually at least a few hundred emails, and I have some friends who have thousands of emails. But the problem is that this was an aberration. This was in about 2011, where Gmail users opened their email account to see that all of their emails were gone, every single one of them. And this was due simply to a Google software update. Luckily, though, most of that data is offline. These are stored on magnetic tape, which most of us don't think about because none of us actually use magnetic tape. But every major digital data company still relies on this technology that was developed in the 1950s. So it required about 200,000 magnetic tapes to restore the emails for all of these users. So as I mentioned, we're constantly generating this deluge of data, and actually most of this data was generated in the last few years. And so we've started to enter what is known as the zettabyte era. So in 2019, not too long ago, we reached more than 30 zettabytes. And it's actually projected that within the next 5 years, we're going to at least double the total data in the world. So this represents all of the digital data in the world. So since I'll be talking a bit about bits and bytes, I just want to familiarize you guys with the different storage schemes. So we start out from megabytes, which is 1 million bytes, and go all the way up to yottabytes. Luckily, we haven't quite entered that many bytes, but we are already in zettabytes. We're even starting to kind of run out of nomenclature for how many bits that we store. So I don't know if many of you remember this, but there's been a lot of talk of space recently, especially with the SpaceX launch. And I still find it very impressive, this photo, the very first photo that was taken of a black hole. This was just last April, so April 2019. And data in order to generate this image was collected across 4 different continents over several days. And so here's a tweet that I really loved that shows the grad student, one of the students who worked on this project to collect all this data. And there she is with nearly 5 petabytes of the data that was needed to compile that single image of a black hole, shown next to an MIT computer scientist, Margaret Hamilton, who actually wrote some of the code that helped put man on the moon. So the thing is, the thing that I love about this image is it's kind of crazy to think that in 2019, they had to ship all of those hard drives. There's this joke that FedEx has a lower bandwidth, a better bandwidth than the internet, even in 2019, because of all the efficiency of sending petabytes of data. And just to give you an idea, that 5 petabyte image was collapsed only into about 180 megabytes. So in the end, it wasn't much, but in order to actually compile it required a massive amount of data. So in a similar vein, this is a picture from the 1990s of Bill Gates holding a CD-ROM when they first introduced the CD-ROM. And there he is sitting on a stack of papers, showing to us that all of the data that could be stored on that CD-ROM is the equivalent of the data on all of the paper beneath him. Now, this is a bit outdated, but the point is that relative to the CD-ROM, this is incredibly impressive, the amount of density that we achieved back in the mid-90s. But most of you, if I were to give you a CD-ROM, most of you probably wouldn't be able to play it on your computer. I have 4 computers on my desk right here, and not a single one of them could even play a CD-ROM. So most of our technology quickly becomes obsolete. Now, we've come a very long way from our first method of storing data back from stone tablets. We've miniaturized, we've increased the density, but every single storage device that man has developed has essentially become obsolete. Now, I mentioned magnetic tape, and this is something that is still widely used, but it's not used by the average person. It's only used as a backup to store our data, and it's really only a fail-safe. And so really all of these technology have become obsolete rather quickly. For any of you who grew up in the 90s, or at least were not in diapers, you'll remember this floppy disk. Much like the CD-ROM, most of us would have no way to even to access the floppy disk, even if you could still recover the data from it. So most of these technologies have become obsolete. Some of them can be repurposed, but the truth is our data generation is outpacing our storage capacity as humans, if we take all of our data here on Earth. But it turns out there is a much better solution. This solution has been optimized for about a few billion years, whereas man-made technology, we've only really been optimizing methods to store digital data since roughly the 1950s. And so DNA is both one of the latest storage devices for digital data, but it is also by far the oldest. So just to give you an idea of the scale of why DNA is a good solution, the main thing is that it is incredibly dense. So if we were to take the DNA in our human body, We have 6 billion base pairs in a single human genome. And we have a copy of our genome in every single cell of our body. That amounts to about 1.5 gigabytes of data in every single cell. Now, we have roughly 30 trillion cells. Someone actually quantified this. And we each have about 30 trillion cells in the average adult. So I don't even put it on the slide here because I run out of space because of all the zeros, but that is a massive amount of data, just to give you an idea of the scale of how much we can fit into DNA in a very small space. So another convenient thing is that every single living thing is a DNA storage device. Nature has been storing data in DNA since the very beginning. So really, storing data in DNA is certainly not new. It's just that recently, since the early 2000s, really only 2010, that we started to turn back to DNA, to turn back to nature, to handle all of our storage data needs. So many people ask, one of the confusing things about storing data in DNA is, well, where exactly are you storing it? What does that mean? So when we say we're storing data in DNA, we store it in synthetic DNA in a tube. I'll show you briefly in a bit, but we do this every day as scientists in the lab. We have ways to write DNA from scratch using chemical synthesis. We can read it and we can store it very easily. So I just want to be clear that it's a long way off before having a literal thumb drive. And some people have suggested the idea of storing family photos in your own DNA so that they can always be recovered. But our genome is pretty useful for making us who we are. And so it's better to store DNA in a tube in a cold, dry place. And so we've seen how well DNA can last by scientists who've recovered DNA from ancient humans. So this is Ötzi. I'm a big fan of Ötzi. He is a mummified man that was found on the Austrian and Italian border in the Alps. And because it was so cold and dry up there, his DNA was preserved. So he dates from about 3300 BC. But scientists were able to recover his DNA such that they were even able to compare his DNA to living humans. And they found that he has living relatives living in Austria today. Now, if I were to hand you a broken cell phone, you'd have a much greater chance of actually recovering information from this ancient human than you would from a cell phone that's only a few years old. So even with the latest technology, data can only be reliably retrieved for maybe tens of years. So I think it's interesting to note that in the 1950s, when computation really took off, when we really started the digital data revolution, this was around the time that we scientists were starting to understand DNA. So this is the first ever photo of DNA. It's called Photo 51. Now it doesn't really look like DNA, but this is the photo that really showed us that double helix structure of DNA. And this was in 1953. Now, soon after, just a few years, this is a picture from 1956. And this is the first ever commercial hard drive being loaded for shipping by IBM. And it could only hold 3.7 megabytes, which these days is about 1 MP3 song. And you can see it's being loaded onto this truck by a small crowd. It would be comical to ship this to someone to store data. You couldn't do much with it. Yet the fascinating thing is that we were able to put a man on the moon with such a limited data capacity. So this thing costs roughly $10,000 per megabyte. And most of us are too stingy even to pay $2 a month to store our terabyte of data. It's really impressive, the scale of the storage capacity that we've achieved just in the last 50 to 60 years. So as I mentioned, DNA has sort of gone digital in the last 10 years. It's been around for billions of years, but it wasn't until really around 2010 that it started to become a viable solution. So I want to give you a rough timeline of the history of the idea of storing data in DNA. So one of the first people to have this idea was American physicist Richard Feynman. And so he had this idea. He never actually went through with it, but he was really one of the first to put this idea into motion, to think about harnessing an existing storage media in nature. And so next up is this guy, Michael Niemann, in the 1960s. And so he also was discussed ways that we could possibly record and store and even retrieve information on DNA. So he took Feynman's idea and actually really articulated how we might do that. But it wasn't until the 1980s, in 1988, with this guy, Joe Davis. I believe he's actually still in a lab at MIT. And he was the first to actually store data on DNA. So the way he did this was actually quite simple. He took this image called microvenus, a very simple image, and he simply translated this into bits. And so basically each pixel in the image is represented by a 0 or a 1. And in the end, it was only 18 base pairs, so 18 A's, T's, C's, and G's to encode this image. This is minuscule. And so he took that DNA and he actually copied it into bacteria. We do this all the time in the lab. It's kind of our living Xerox machine in the lab. We use E. coli in order to copy our DNA. And so he copied this into E. coli and was actually able to store the microvenus image in DNA. Now, he didn't actually recover the data, but it was a nice proof of concept of how we could actually do this. So that was again in 1988. Just to give you a rough timeline, since then, not a lot was done in those intervening 10 years. But in 1999, there was a DNA encoded encryption of the message June 6th invasion in Normandy. And it wasn't again until roughly 2010 that the idea of storing data in DNA started to really take off. And within those few years after that, there were some incremental advances in the amount of data we could store. So for example, in 2012 and 2013, roughly 0.8 megabytes were encoded, but they were recovered with errors from the DNA. So scientists were able to actually store this data in DNA, but they weren't able to recover the original files without errors. And so more recently, from 2015 on, we've had some advances that include being able to store data and recover it without error. Also to actually use random access. So when you store data in DNA, you can store it in a tube and you have to sequence the whole thing in order to get your data back. And so researchers showed that you could actually randomly access different files using DNA. So it's a tedious process, but it can be done. So you don't have to store all your data in one tube and sequence all the data. You can really target and sequence a subset of this data. And so we can see that the scale of the data stored started to increase. So back in 2016, roughly 22 megabytes was encoded and recovered without any error. And back in 2017, my previous boss and I published a method that allows error-free storage at a density of 215 petabytes of digital data on 1 gram of DNA. So I'll talk briefly about our study in a moment. So soon after our study, and there were many studies before us, it certainly wasn't our idea to do this, but there were many other studies following with some incremental advances. I certainly don't list all of them here, and this may be the last presentation where I can actually list the significant milestones in DNA data storage on a single slide. So in 2018, we were able to encode and recover without error 200 megabytes of data. So this is quite an increase in scale of digital data that we were able to store and recover. And just last year, a company in Boston was able to store all of Wikipedia, which amounted to about 16 gigabytes compressed in DNA. But of course, they didn't disclose how much this costs, which I'm guessing was astronomical. And so I'll get into the cost of actually doing this in a bit. So we were also able, scientists were also able to enable end-to-end automation of DNA storage. This was Microsoft researchers and at the University of Washington. It was a small message, it was only 5 bytes, but it was a nice proof of concept of really an end-to-end of where you take your digital data, you encode it in DNA, you store it, and then you recover it by sequencing. And so they did this all in one end-to-end device. And more recently, in 2020, there was a method called the DNA of things, where researchers actually took synthetic DNA, so they encoded their digital files in DNA, and then they actually were able to encapsulate this DNA and mix it in 3D printing material. And this allowed them to actually print, for example, the instructions in a digital file into a 3D printed object, so that you could actually break off a piece of that 3D object and sequence to get that data out. And so you can find this if you Google DNA of things. They stored data in a 3D printed bunny, and they were able to show that they could break off a piece of the bunny, extract the DNA, sequence it, and get all their files back. So I want to go back to that first image of DNA, just as a sort of proof of concept to show how we might actually store this digital file in DNA. So imagine I take this file. It's made up of zeros and ones, or bits, short for binary digits. And so if I wanted to store this photo of DNA in DNA, I simply have to convert all of those zeros and ones into A's, T's, C's, and G's. So as I mentioned, in the lab, we are able to write, store, and read DNA. Scientists do it all the time. We use synthetic DNA in order to clone new sequences into E. coli, for example, into different cells to genetically modify them. We read the data by sequencing it. The thing is, these processes aren't as straightforward as they seem. While they are very easy to do, writing DNA is incredibly expensive. One huge advantage, though, is that storing data in DNA, it just requires a cold, dry place. You don't need any special equipment to store data in DNA. You could store it in your wine cellar, in your refrigerator, and it can last hundreds, even thousands of years. As I showed through the recovery of the DNA from Ötzi the Iceman. And reading has actually become very affordable. Back in 2003, when the first sequence of the human genome was published, it took about 10 years and billions of dollars. Now, here we are in 2020, just 17 years later, we can sequence an entire human genome in a matter of hours for a few $100s. So sequencing costs have dropped enormously in a pretty short time. So I want to explain a little bit about our study that we published back in 2017. As I mentioned, we weren't the first to store data in DNA, but we did achieve through our study back in 2017 the highest density to date. And while DNA is already dense, we really wanted to explore the limits of DNA storage. And so to do this, we used a metric called the Shannon capacity, named after Claude Shannon, a really remarkable guy. He was the father of information theory. And I'm just showing this simple equation because this is sort of what we used when we were trying to understand the limits of storing data in DNA. And so this is called the Shannon capacity, and it tells you how to calculate the maximum rate that information can be communicated over a communication channel. So this is information theory, where we're kind of mixing DNA and information theory here. And so this shows how information can be sent over a communication channel with 0 error. And so if we treat DNA like a communication channel, bear with me, when we're sending digital data, over a server, if you're streaming a file, downloading a file, this is not very different from the process of storing data in DNA. As I showed, we simply take your digital data, we transfer those zeros and ones, we convert them into A's, T's, C's, and G's, and then we sequence it to get it back. So there's a much higher latency here. It takes quite a while to write the DNA and to read it back. But we still wanted to explore the theoretical amount of information we could store in a single nucleotide, which is theoretically 2 bits per nucleotide. So I want to give a simple example here of how we could actually store data in DNA. Now, it's not quite as easy as I showed. You don't simply convert the 0s and 1s to A's, T's, C's, and G's. And that's because DNA has some biochemical constraints that make this difficult. And also, in order to actually get your data back, sequencing can actually introduce quite a bit of errors. So we need a more efficient way to do this. And so suppose I want to encode this information using digital data. So this would require 3 times as many bits if we actually wanted to store this. And this is really highly inefficient. So we can use error correcting codes that actually can help reduce the errors when we actually store digital data in DNA. And so when we're exploring the Shannon capacity, we're really trying to understand, to try and find a balance of the minimum amount of extra information in order to get the maximum data back. So if we want to send this simple 3-bit message without any error, we have to add some padding to it. And this gets highly inefficient. And so there are ways that we can get around having to add these extra bits known as error correcting codes. So one method we use is called fountain codes. And these are capacity approaching codes known as Luby transform. These were developed in the late 1980s. And they're called fountain codes because you can take your digital data and transform it into an unlimited number of encoded chunks known as droplets. Now in these droplets are your 0s and 1s. So you take your original data and you specifically encode your data into these droplets. So according to a specific distribution, you'll select different chunks of your data and separate them into droplets, such that in order to recover your data stored using this Luby transform, this fountain code, you simply need to retrieve slightly more data than you stored. And now most communication channels are sort of a 2-way communicator where the receiver needs to communicate with the sender saying, yes, I got the message. But with fountain codes, they're more resistant to error because you simply can send your bucket of droplets. And as long as you send slightly more data than your original file size, you can recover the original data without error. So when we're looking at the Shannon capacity of DNA, it's not quite the same as just simply storing 0s and 1s. We have 4 options per nucleotide here. So that's why we can get 2 bits per nucleotide. So this is the theoretical maximum. But the reality is we can't actually store 2 bits of data per nucleotide. And that's because DNA has some constraints that can introduce errors when you're writing or reading it. And so take this sequence, for example, a simple string of DNA, and it can be quite challenging if you have long runs of the letters C's and G's. And for those of you who remember from high school biology, this has to do with the fact that there are 3 bonds. And so these C's and G's can make it quite challenging in the writing and the reading process. We also, when we're trying to store data in DNA, we specifically avoid these long runs of a single letter. So here you see an example of a long run of C's. We try and keep those segments down to only 3 or 4 C's in a row, or 3 or 4 A's in a row, because this can, again, introduce errors in the DNA synthesis and the DNA sequencing process. So if we take into account those biochemical constraints, we can only actually get, which is not bad, a limit of 1.98 bits of data per nucleotide. But once we have to add, again, as I mentioned, when we were storing that 010, we have to add some padding in order to correct for errors. And so that gets us down to about 1.83 bits per nucleotide. And our net information density, once you take into account the size of the DNA sentence, so we're limited by how long the DNA is that a DNA synthesis company can print. And so this will further reduce what we call the logical density to about 1.6 bits per nucleotide. So that's really the theoretical maximum of what we could achieve. And so the fun part of storing data in DNA is deciding which files to encode. So in our study back in 2017, we encoded one of the smallest operating system known as Calibri, one of the first movies ever made, manuscript by Claude Shannon, an Amazon gift card, a zip bomb for, just for fun, and the Pioneer spacecraft plaque, which is supposed to show to extraterrestrials where we are in the galaxy and what typical humans look like. So we simply took these digital files, we created a tar archive and compressed the data. And then we applied our fountain code method, and we created literally just a spreadsheet of A's, T's, C's, and G's derived from these files. And within about a week and a half, we had our data stored in this tube. So that was roughly 2.4 megabytes of digital data in DNA. And that's the actual tube that we got in the lab. So as I mentioned, we can relatively easily write, store, and read DNA. But there are some limitations, and I want to be honest that commercializing digital storage in DNA is going to take some serious engineering work, especially in reducing the costs of writing DNA. Another limitation of DNA is that if you were to access your digital data on a hard drive, you're simply reading it in real time, right? If you're reading DNA, if you want to read your digital data off of DNA, you have to sequence your DNA. So you take that tube, it typically comes as a dried up pellet, you add some water to it, and then you have to take some of that DNA out to sequence it. So really the process of reading your data destroys the data. And this tube was quite expensive to generate. But luckily, we have a solution that we use in the lab every day called the polymerase chain reaction, PCR. And so this allows us to essentially have unlimited copies of our data. So we could take that original tube that the synthesis company sent us, we can dilute it with some water. We add some enzymes that will copy the data for us. So we can essentially copy our data an infinite number of times. Now, you might wonder this, I mentioned that the process of copying and reading and writing DNA can introduce errors. And this happens in our cells. But of course, in our cells, there's enzymatic machinery that can fix those errors. Now, we use enzymes that are supposed to reduce these errors, but they certainly do happen. And so we actually tested the maximum amount of data we could retrieve after copying the data trillions, literally trillions of times. And by using this process, we were still able to recover all of our data without any error. And so we were able to generate 10 to the 15 possible copies of that original library. So we copied it that many times, recovered the data without error. And so, as I mentioned, we are in the zettabyte era here. And so we were able to show in our study that we could store about 215 petabytes of digital data in a single gram of DNA. So what this means is that if we kind of cut the difference between 2019 and 2020, and we say the world's data amounts to roughly 35 zettabytes, that means that if we stored all the world's digital data in DNA, it would weigh about 160 kilograms. So that's an incredibly impressive density. Now, I want to mention some of the caveats of storing data in DNA. I mentioned that it's incredibly dense, it's incredibly durable, but we need a bit of a reality check here. To synthesize those 2 megabytes of data cost us $7,500 US. Now, I don't know anyone willing to spend that much on storing their data, whether it be precious photos or important documents. But this is something that really needs to come down, the cost of writing DNA. And there have been some advances in the past few years in reducing costs and also improving things on the computational side. So we can do what's called a messy synthesis, where we can tolerate lots of errors in the writing process and handle those down the line. So DNA is about 5 orders of magnitude, give or take, more expensive than traditional storage technology, but it's more than 5 orders of magnitude more dense. So there's some really important trade-offs here. And while storing real-time data in DNA is at least, I'd say, 5 to 10 years off, storing low-touch archival data is, I think, a real possibility in the very near future. So I mentioned writing DNA too. This process is nearly 200 times slower than storing digital data on a hard drive, for example. But the trade-off there is that this data can be stored for hundreds to thousands of years. So again, it can be very useful for low-touch archival data. And our reading bandwidth, so actually being able to read that data back as we need it, is limited simply by the number of sequencers in the world. So really our sequencing capacity. And roughly 2 years ago, we determined that the world sequencing capacity was roughly 1 petanucleotide per day, which amounts to roughly 25 terabytes of data that you could recover from DNA throughout the world in 1 day. So really, what is DNA's competitor? I've shown that it's way more dense than and outlasts any man-made device. But right now, as I mentioned, DNA's main competitor is actually magnetic tape. So I've never actually used magnetic-based tape for storage, but as I mentioned, companies like Google, Facebook, they do. And this is still the cheapest, most reliable way to back up critical data. As I mentioned, magnetic tape was first used to store data in the 1950s. This was around the time of that first commercial hard drive and the first, really when we first started to understand DNA. So I really I love that our knowledge of storing digital data and of understanding the structure of DNA has sort of co-evolved, although we've put way more effort into advancing storage media for digital data than we have to put into existing DNA in order to store data. But that has changed in the last 10 years, and we've made some major advances in the last 10 years alone. And eventually the cost per gigabyte will outpace magnetic-based storage. So DNA will get there eventually, but it's largely right now an engineering problem. So maybe as early as the late 2020s, maybe within the next 10 years, DNA storage could outpace magnetic tape. So I think I've convinced you of the scale of the data storage, but I just want to compare some of the top of the line man-made devices right now that we use to back up our data. Now, in order to actually store all of this data, you would need roughly 350,000 100 terabyte flash drives to store all the world's data, and roughly 100,000 300 terabyte tape cartridges. So this certainly doesn't compare to that 160 kilograms of DNA. This would take up a massive amount of space, and it does. So I was looking for some other man-made storage solutions, especially things that are more portable. And one of the densest that you can actually transport your data on is a 2 terabyte thumb drive. But this thing costs roughly as much as a MacBook. So I don't know about you, but I've misplaced many a thumb drive. So this is really not a very viable solution. And if we actually compare the density, I showed that we can store 200 petabytes of data in 1 gram of DNA, that it can last hundreds of years as long as we keep it in a cool, dry place. Now, -20 is extreme, but it really can last hundreds to thousands of years below -20 degrees Celsius. But if you keep it at, say, 4 degrees, it can easily last tens of years. Whereas most man-made storage devices, the storage density just cannot compare, and they last 5 to 10 years at best. If you try and think of a device that you have that's lasted more than 5 years, that you still use to access your data, I think most of you would have a hard time coming up with something. So again, magnetic tape, the biggest competitor to DNA storage, data can really only last 10 to maybe 50 years. You still have to keep this tape in a safe place. It can be very temperature sensitive. It's much faster than reading and writing on DNA, but it still takes time because you have to go retrieve the tape. You have to read it onto a device that actually has some computation power. And of course, the biggest caveat here is that storing data on magnetic tapes only costs about 2 cents per gigabyte, whereas DNA costs roughly $10,000 per gigabyte. But as I mentioned, we've really come a long way in reducing the cost of reading DNA, of sequencing DNA, since the early 2000s. And we really have put very little effort since then into reducing the cost of writing DNA. And that's because scientists need that DNA synthesis to be perfect for our experiments. But when it comes to storing data in DNA, we don't necessarily need perfect synthesis. And so scientists are currently working on using enzymatic synthesis to reduce our reliability on chemical synthesis, which can actually also be quite detrimental to the environment. But if we can move again to a solution from nature by harnessing existing enzymes to write our DNA and allow tolerance for errors, I think that within the next 10 or so years, that DNA storage will really become viable. So the other example that I've mentioned is that every single device, every single man-made device has become obsolete. And this exponential growth of reducing the cost per gigabyte, it's not going to last forever. We've really already reached the density that we need with DNA. And given that DNA encodes all living things, it's as long as we are around, DNA will never be obsolete. So as a storage technology, we really don't have to worry about obsolescence. So just to sum up, this is showing you the progress that was made in the last 60 years in man-made storage technology. So again, that's that first hard drive being loaded in 1956 that stored at roughly 4 megabytes. And now we can have 200 gigabytes, probably even more now, the size of a quarter. So we've come really a very long way in man-made devices in the last 60 years. And we've also come a long way in sequencing technologies. This is one of the first sequencers, roughly the size of that hard drive. And now we can use a sequencer that is, you can see a coin here for scale, but sequencers have become miniaturized, and you can even carry a USB sequencer with you. Astronauts have even brought this sequencer into space. And so we've really come a long way in man-made technology for storing digital data and also in sequencing. And once we can focus on writing, we will definitely be able to make DNA storage a reality. So in the time that we first took that first image of DNA 60 years ago, we've learned how to read DNA, how to write it, and even edit. We can now edit DNA. So really, we may be storing our most valuable information in DNA sooner than you think. Thank you.
Dr. Dina, we have limited time now, and Mr. Daria, if you can proceed for the Q&A, please.
Thank you, Dina, for such a talking on this breakthrough stuff, how to store data in DNA. And you're right, the way you have shown your graphics and presented in your presentation, big data is not sustainable. And I think in last 2 months, the way Corona has attacked on us, we realize that no more we can really hurt nature, and we need something which can be nature friendly. And DNA is, of course, one of the nature friendly technology to really store DNA. data. And this could be a big substitute for big data storage. And there were some questions around this. One of gentlemen has asked a question: when big data is already there, why to use DNA storage? Of course, you have answered, but I think that will sum up your entire talk.
Yeah, absolutely. I mean, DNA storage it has a lot of pitfalls, right? It's very expensive to do. But as I mentioned, we simply haven't put the effort until very recently into actually making this a reality. And of course, storing data in DNA can also be harmful to the environment. We still need the sequencers. The process of writing DNA right now is very wasteful. We generate a lot of waste products through the chemical synthesis. But I think there are solutions that will really make this a reality very soon. And the fact is that every email that we send, every photo that you save, every tweet you tweet is detrimental to the environment. And DNA storage is not really a solution for that because of the high latency, but we can actually at least free up some of that server space and store some of our most secure, precious data in a reliable way.
So in a way, what you're saying that though at this stage, DNA storage is very nascent stage, it still has not reached to the commercial viable proposition, we can still store it, some data which could be for archive purpose, which we are not using very frequently.
Exactly. I mean, one important thing to think about is we've learned a lot about human history through human artifacts, through writings on caves, through the Rosetta Stone. But our generation and future generations won't have this. We simply don't have non-digital artifacts. Most of humanity now is digital. All of our scientific discoveries, our financial records, our family photos, most of us don't even print photos anymore. Everything is digital. And I mean, imagine on a small scale losing all of your own data. It would certainly be a personal tragedy, but imagine all of humanity losing our digital data, starting from scratch. So I think it's a really fantastic option for archival data to store some of the data that's most important for humanity. And as a scientist, the first thing that comes to mind is storing all of the scientific publications, because that's how we make incremental discoveries is we build upon existing research. So I think that's a very good solution for it.
Yeah, and that's in fact a very natural way that DNA stores all the information about the humanity and everybody, everything. Why can't really we retrieve this information in DNA and put it? And in fact, this big data, Amazon or Google, they are having roughly around close to 1.5 million servers, and which is costing billions of dollars and so much of power usage, so much of damage to the nature. And I was amazed by this analogy, which the example you gave, that whatever the data we have created in this world, that can be stored in only 160 kg of DNA. And that's something very amazing stuff, right?
Yeah, you can fit back of a small truck.
Yeah, maybe just size of weight of 3 human beings, right? Or maybe two human beings, right? And that's incredible, right? Now, there's some questions Aditya is asking. What is the process of writing in a DNA? So there is a process of writing, storing, and then retrieving. So where is the bottleneck, and how do we really write on the DNA? What is the process?
That's a good question. So as I mentioned, the bottleneck right now is really the writing. I showed that graph of sequencing cost dropping, and sequencing has become highly affordable, and sequencing is the way we retrieve the data. To actually take your digital data and write it in DNA requires enzymatic synthesis. So we literally send a spreadsheet of A's, T's, C's, and G's to a synthesis company, and they will print. It's much like an inkjet printer, except that it's a chemical process that will literally just print the strands of DNA in the order that you specify. So the chemical printer will print an A, followed by a G, followed by a C, and so on.
Okay, so the current problem is typically writing, and you gave the solution about retrieval. So where are the bottlenecks in retrieval process?
So as I mentioned, it really depends on our sequencing capacity. So roughly 90% of the sequencers in the world are Illumina machines. So if we were to take all of the Illumina machines in the world, we would be able to recover roughly 25 terabytes of data per day. So our reading capacity is limited by the availability of sequencers. And normally when we in a lab want to sequence data, there's always a queue to try and fill up the sequencer and get space on the sequencer. And so we're limited by really by the physical number of sequencers.
So that if we really see the history of all the storage devices over the years, the cost of storage, retrieval, everything really goes down when the economy of scale really comes into the picture. Hopefully people will really take a call on this and they will start investing and bringing focus on this method as well. There was another question somebody has asked about quantum computing. How does is quantum computing will be able to resolve the issue of data storage, or how DNA actually compares with the quantum computing?
So this is a great question. This is actually a huge advantage of DNA over magnetic tape. Magnetic tape is it's kind of dumb, right? It's a media that you store zeros and ones on. We can actually use DNA to compute. So not only as a storage media, this is kind of, I think, the next stage in the digital data storage and DNA field, is to really start harnessing DNA to actually do computation. So I think DNA computing is not that far off. And again, this can help with our storage problem for sure.
Wonderful. Umesh Krishna is asking, DNA being biodegradable and disintegrates at a higher temperature, how is a reliable media for storage for long term? Ozzy was lucky as he was stuck in ice. We also stuck this storage into ice.
Yeah, no, it's true. Like I presented this idea when I was in Rio, Brazil, and they called me out on this. Well, we can't just store DNA, somewhere in Brazil. It won't last more than a few years, and that's absolutely true. But again, we can store data in DNA using zero electricity if we simply store it somewhere cold and dry. But again, man-made technology is also sensitive to temperature and moisture. So this is not new. And DNA, we've seen, is able to last tens of thousands of years. And we have yet to see a man-made device that lasts more than tens of years.
So yeah, I remember there was a time when even the normal PCs were kept into AC, right? While we were doing our engineering in 95 and 93, we used to really enter into the computer lab, which was completely air conditioned, right?
Absolutely.
So hopefully someday we can really build some storage devices which could have this low temperature. We have some interesting questions people are asking. Do we consider encrypting the data while storing in DNA?
Yeah, so you can encrypt data in the same way you would encrypt digital data. Anything that you can do to digital data, you can do with data stored on DNA. Because really it just comes down to preparing those files, whether it's encrypting, adding error correcting codes. Once you have your final file, all those zeros and ones, it's really just a matter of converting the zeros and ones to A's, T's, C's, and G's, as I showed. So 0, 0, for example, becomes A, and so on.
So Pooja is asking a question: if a pen drive or any kind of memory chip, the computer knowledge is enough to handle that data, but in this now storing data in DNA is creating challenge to access that. How to overcome this?
So to have if I understood, to have a thumb drive, a DNA thumb drive?
So what I could understand, like the knowledge in terms of really retrieving the data and accessing the data is still, it's specialized. It cannot really be given into the common man's hand, just like a flip, say pen drive, right? Which will take years. Yeah.
Right. Yeah. So the closest that people have gotten really is the researchers at the University of Washington when they encoded 5 bytes of data in an end-to-end solution. It's still quite big, the footprint of all of the chemicals and the bottles and the sequencer. But that sequencer I showed on my last slide, it's like a very large thumb drive. So this has certainly been explored and there was a proof of concept, but being able to miniaturize that, as I mentioned, is really, it's become more of an engineering problem.
Right. So another question by Rahul, DNA has a feature called mutation. Will mutation affect information at a time of retrieval or storage of data?
Absolutely. So mutations can occur in every stage of the storage process. This also happens in our cells. Our cells accumulate mutations throughout our lifetime. And there are actually enzymes that can go ahead and correct these mistakes. But when you're dealing with synthetic DNA in a tube, there's not really a way to correct the errors. And so we really, most of the solutions lie in using these error correcting codes, of course, and also really handling the errors on the computation side. So really finding methods, computational methods that allow us to handle those mutation errors. And so in our study, we actually did this. We copied the DNA trillions of times, which inherently introduces errors. And we were able to recover the data without any error.
So thank you, Dina. So we have other speakers lined up. So I think, hello, Usma, Dr. Usma. So I think this is very good solution, which could really help this data hungry society to store in a data DNA. Thank you so much, Dina, for joining this session.
Have a great evening.
You too. Bye-bye. Dr. Fayad, welcome to you again.
Thank you so much, Bhupesh. Good to be here. Wish I were there in person, but hello from San Francisco. And my co-speaker is in Boston.
Thank you so much. Thank you so much for being with us.